Hi everyone, I'm Janine Cavender-Bares at the University of Minnesota, and I'm going to talk today about how the reflectance of photons from the sun from plants reveals a great deal about their phenotypes related to genetic and phylogenetic information and ecosystem functions. Much of the work I'm talking about is based on a long-term collaboration with many of the colleagues you see here and has been funded by the National Science Foundation and NASA. There are four major areas I'll cover. I'll first start with some background on the sun's electromagnetic spectrum and what reflectance spectra are. And I'll talk then about phenotypic information that spectra reveal in relation to genetics or phylogenetic history, and then how those spectra relate to ecosystem processes. And then finally, at the end, I'll talk about a specific project that we're working on to use spectra to detect plant disease, oak wilt in particular. It's an important disease that affects oaks in Minnesota and throughout North America. The basic idea is that light interacts with plants in a way that reveals a great deal of information about plant function and the chemistry, the anatomical structure, and many attributes of phenotype that are then linked to phylogenetic genetic information. And so this spectrum, these two spectra that are represented here in this image from two different plant leaves, you can see how they show somewhat different patterns. And that information can be hard to see right off from a graph like this, but because there are hundreds of wavelengths of information in that spectrum, it's very high dimensional information about plant phenotype. And so this is just a reminder that we're talking about energy from the sun's electromagnetic spectrum, and those incoming photons that are reflected off surfaces, and in this case, plants. And we're really talking about the region that spans the visible to the shortwave infrared from about 350 or 400 nanometers to about 2,500 nanometers. And this is a typical reflectance spectrum. On the x-axis is wavelength, and the y-axis is reflectance. Here's the visible region where your eye can see, and there is a lot of absorbance there, so less reflectance. And then in the near infrared, there's more reflectance and less where water is absorbing. And so you can see these troughs where in the reflectance spectrum where water absorbance is occurring. And so in general, pigment absorption is the dominant process in the visible region. Scattering is the dominant process in the near infrared, and water absorption is important in mid infrared. And many chemical compounds are absorbing in different parts of this spectrum, and they each have distinct reflectance signatures. This image, which is generated from former postdoc Dudu Mireles, just gives you an idea of how much information you get from having reflectance in many, many wavelengths. On the left, you see this image just black and white. And when you just switch to 3 color bands, the red, green, and blue, this markedly increases the information we can extract from a picture. And that's with our eyes that can capture those 3 bands. But when we go beyond the visible region and can capture hundreds of wavelengths of reflectance information, now we get all kinds of information that our eyes can't see but that turns out to be quite important for understanding plant function and phenotype. Now this spectrum is showing again wavelength on the x-axis, and it's showing what we would call full range spectrum from 400 to 2500 nanometers. And then the blue is reflectance, but the green here is transmittance. So light is either reflected, absorbed, or transmitted. And the light that's neither reflected nor transmitted through the leaf is absorbed by the leaf. And you may be familiar with looking at absorptance. And in this case, we see an absorption spectrum. On the y-axis now is absorption by different chemical compounds. On the x-axis is the wavelength, here just showing the visible region from 400 to 700. And this is the typical spectrum of chlorophyll A, and here we have beta carotene. And these are compounds, these are common pigments in plants. What I'm talking about today is the reflectance spectrum of plants. And this is from a figure from Sean Serban and Phil Townsend, showing the typical plant spectra from different plant leaves or canopies. And maybe these are dried leaves that don't have water absorbing, or a soil in these darker colors. So here we have litter, wood, soil. And the shapes of these spectra reveal different things about pigments, water content, and scattering, nitrogen, cellulose and lignin, sugars, starches, and proteins. And I'm not going to talk about the methodology today, but there's a wide range of different sensors that are used and that can be used on different platforms to either detect at the leaf level or remotely sense reflectance spectra. We'll start by talking about an experiment that we did with one species. This is in Central America. This is a common garden of different genotypes of Quercus live oak species in Honduras. And this was part of a longer study looking at the phylogeographic patterns and genomic variation in this species. But we wanted to see if we could use spectra to differentiate genotypes. And so we measured leaf level spectra in this experiment. And we also ended up doing this in many different species of oaks, which I'll show in a minute. But there are two main points from this work. And the first is that spectra provide a lot more information than do individual traits that we might predict from spectra or measure in some other way, in terms of our ability to differentiate genotypes or species or phylogenetic lineages. And this is from a paper in 2016 that I'm happy to send. The second point is that it's easier to detect higher order lineages than genotypes within species. And so the broader the lineage, the easier it is to differentiate that. At least that's what we found with the oaks in this particular study. We use partial least squares discriminant analysis, which is a form of machine learning. And we detected both traits and the spectra and determined which regions of the spectra were informative in differentiating genotype, species, or clades. And simply by having many, many more informative regions for spectra, that high dimensional information, there's more to work with in classifying genotype, species, or clades. So we also measured spectra on oak species in a common garden, and this is part of a longer term study looking at the phylogenetics of the oaks in the Americas and globally, and that work's been led by Andrew Hipp and has been very exciting to be involved with. We We again measured spectra on different species of oaks and used the phylogenetic information to put all these species into a phylogeny. The colored circles represent clades, phylogenetic lineages. And what's shown here is the first principal coordinates axis for the spectral information, which shows high phylogenetic conservatism and is different among different clades, which you can sort of see here. We also predicted traits from those spectra, and some of those were phylogenetically conserved, as you can see in red, and some of them were quite labile, as you can see in gray. We looked at different parts of the spectrum for all those different species and have found that the shortwave infrared region was the most phylogenetically conserved. And I'll go more into that subsequently. But the idea is that there's information in here that your eye can't see that's tightly coupled to phylogenetic information. And in general, this is showing what I mentioned before, is that with the classification accuracy using spectra to differentiate genotypes or populations within a species was lower than our ability to differentiate, identify species, which itself wasn't quite as good as our ability to differentiate clades. And all of these could be significantly differentiated, detected from spectra, but our highest accuracy was differentiating phylogenetic lineages from each other, which is quite interesting. And I'll come back to that shortly. In another study, we were working with aspens, 2 parental aspen species, Populus grandidentata and Populus tremuloides, and then we were trying to determine if a population in Niobrara River was a hybrid, and we did the genetic work for that, but this is using spectral information and functional traits to differentiate the hybrid from the parental species. So in purple is the hybrid, and the red and the blue are the 2 parental species. And what's interesting here is that when we use the full spectrum, it was very good at differentiating the parental species and also differentiating the hybrid from the parental species. In a study at the Cedar Creek Ecosystem Science Reserve in the diversity plots, which we call BioDiv, that Dave Tillman set up several decades ago, we measured leaf level spectra on plants in that experiment and found that the spectral distance, so if you sum the Euclidean distances between all the reflectance and all the wavelengths and relate those in a pairwise fashion to their functional distance in terms of multivariate functional traits. In general, those pairwise relationships are all tightly associated with each other for each species. The same is true for phylogenetic distance, except in some cases where the leaf shape is rather complicated, and in those cases, we did not get positive associations. But in general, spectral distance was highly associated with functional distance and phylogenetic distance between species. And using the same classification method, partial least squared discriminant analysis, Anna Schweiger was able to significantly differentiate all the species in that system, except sometimes the grasses got confused with each other. And again, when species would get misassigned, it was largely within the same lineages. So again, you can see the strong phylogenetic signal that's coming through in these spectra. Now I've mentioned but haven't really explained that spectra predict functional traits with high accuracy, and we already know standard methods for measuring pigments is using spectroscopy, often in aqueous medium, but you can also do it with reflectance on dried tissue or fresh tissue. And here we're using full spectra and using statistical methods, in this case, partial least squares regression, to use the features of the spectra that best predict the functional traits that we measure in some other way, and then we predict those traits using these statistical machine learning methods. And so we develop a series of models for the spectra that we can then apply when we measure spectra in new individuals, new plants, as long as those models are robust. And this turns out to work quite well. And Xihui Wang, former postdoc in Phil Townsend's lab, then used these models to map the functional traits of species in that biodiv experiment at Cedar Creek. And here's just a visual of mapping traits across the experiment. And you can take a look at this study if this is of interest. And that's actually related to this imagery that's now in the Bell Museum. It's a visual representation of hyperspectral imagery, so these reflectance spectra of these plots in the biodiv experiment that can be represented visually based on the information content in those spectra in all of these plots. So this pretty obvious linkage between the high dimensional information contained in spectra and phylogenetic information led us to start a working group through the National Institute for Mathematical Biology and Synthesis, and have people bring together their leaf reflectance spectra and conduct a series of analyses and phylogenetic modeling approaches to see how well leaf spectra reveal phylogenetic information. So this is the group that came together. And I already mentioned Dudu, Jose Eduardo Meireles, who was a lead author of the work that came from this, and you can find that study here. And so there was a part of this project that involved everyone bringing together spectra, but they were measured with different instruments, and so the sensors were somewhat different. There was an initial part of this that involved translating spectra from one platform to a common spectral signature so that they could all be interpreted in the same way. And you can read about that in the paper. What I'm going to talk about here is then once we had a common set of spectra from all the participants in the study measured on whatever instrument they were using, these were all at the leaf level, and they were collected from these different regions here. The phylogenetic and Dudu put together the very careful phylogeny for all these species. And what's shown here are mean spectra for those major lineages that are colored here. And then up here, those major lineages are shown, and you can see the phylogenetic signal, in this case using Blomberg's K, for all the different wavelengths, reflectance in all the different wavelengths. So the signal for those values for each major lineage. And what's interesting is that there's a high amount of phylogenetic signal in many, many parts of the spectra, but it's different for different lineages. And so you can see the rosids have high conservatism in spectra all the way across the spectrum, except for certain parts of the shortwave infrared. The monocots are similar, but there's more lability in parts of the shortwave infrared. The Magnolidae, have other places that show high lability or not conserved. And then the gymnosperms don't seem to have much conservatism in the visible or near infrared region, but high conservatism in the shortwave infrared. And then we see a contrasting pattern with the non-core eudicots. And then here are the asterids. And you can look at this at any scale, any phylogenetic scale, down to families, genera, species, and so forth. But the basic finding here is that phylogenetic information and spectra are tightly coupled, such that the spectral information reveals a lot about the evolutionary history of seed plants. And Dudu did several different kinds of modeling approaches. He used radiative transfer models, which use type of physical model that uses key functional attributes of plants to generate spectra. And Dudu took one of those traits at a time. You can see them here: leaf layers, chlorophyll, carotenoids, water, leaf mass per area. He took one trait at a time and allowed it to evolve based on an evolutionary model. And that's what you're seeing simulated here. And then spectra were generated at each step through that simulated evolution. And then he was able to run the models in the backwards direction and look at the reconstructed traits from those different spectra and make interpretations about the evolutionary history from those. And so this is just an idea of one thing that can be done with spectra. If you have spectra for a wide variety of plant species, you can then, using evolutionary modeling approaches, determine whether their phenotypes are highly convergent or constrained or appear to have followed, are more consistent with patterns of drift. And this is again showing partial least squares discriminant analysis, in which Dudu shows how well that spectral information can differentiate these different major lineages. And it's with very high accuracy. The diagonal is showing the probability of getting the identity correct. And these show the probability of assigning it to some other lineage. And you could do this again for families or genera or species, and the accuracy is quite high. Gerard Sapes, a postdoc right now, has been using spectra from tree canopies out at Cedar Creek Ecosystem Science Reserve. And these are individual species, and the dark colors indicate high accuracy in differentiating one species from another based on remotely sensed reflectance spectra. So this works not only at the leaf level, but also at the canopy level with those same kinds of sensors, but on airborne platforms. So we going to move now into talking a bit more about remote sensing of spectra, linking them to diversity, plant diversity, and then to ecosystem processes. And so in that same biodiv experiment at Cedar Creek, you can see that there are a whole series of plots. In each one of these plots, airborne imagery will get a number of different pixels. And I've just tried to show a cartoon of that here, and then you can see each pixel will have a different reflectance spectrum. With airborne imagery, some parts of the spectrum you can't get signal because of the atmosphere, and so those are removed from the spectrum. And so one thing we were trying to do is see if we could detect plant diversity from airborne platforms. And ultimately, we had this idea that if we could detect diversity and composition, vegetation chemistry, and biomass or productivity, that might tell us something about below ground processes and ecosystem functions that may ultimately allow us to link, to gain an understanding through remote sensing of how biodiversity and ecosystem functions are related to each other, and to learn something about what might be going on below ground based on what we can detect from airborne platforms. So this was sort of a conceptual framework for thinking about that. And we worked in two different grassland biodiversity experiments, the biodiv experiment at Cedar Creek that I've been talking about, and another one at Wood River in Nebraska, where the plots are much bigger, the soils are much more fertile. And so we put in, so there were subplots within each of these very large plots. And in this case, there's 100% cover in every plot, whereas in the biodiv experiment, because of weeding, some plots have exposed soil. So there's some big differences in these two experiments, and also there are big differences in soil type and fertility as well. So in the biodiv experiment, it was possible to detect diversity using spectral information, and we just used a simple measure of spectral variation, the coefficient of variation. but it really only worked at very fine spatial resolution. It didn't work very well with airborne imagery. So we used a tram that would be a lot like a UAV to have the optical sensor above the canopies. Now in Wood River, where the plots are much bigger, it actually worked quite well. So this is with airborne imagery. This work was done by Ran Wang, and the airborne imagery work was done by Hamid Gulizadeh, University of Nebraska, with collaborator John Gammon. And so Hamid found that the coefficient of variation from spectra nicely predicted different metrics of plant diversity in the Wood River experiment. But it was trickier at BioDiv because of the bare soil. Now, Anna Schweiger showed that we could take spectral information and calculate spectral diversity. She used a different method to calculate spectral diversity, but nevertheless, when she measured it at the leaf level and then calculated spectral diversity of plants from that, it very nicely predicted productivity in the same way that Dave Tillman had previously showed species diversity predicts productivity, or that Mark Cadat had shown phylogenetic information predicts productivity, or that we had previously shown that functional diversity predicts productivity. Now Anna's showing that spectral diversity predicts productivity. And that was true at the leaf level, but then she also used the spectral information that was remotely detected from Ran Wang's work, which there was no information about species or anything, and using the fine spatial resolution, or the high spatial resolution information, that also nicely predicted productivity. So going back to these two experiments, we had the idea that there would be different limiting factors in each system, and so different drivers of soil microbial processes and soil nutrient availability below ground. And in BioDiv, we thought, well, because productivity is actually rather low and there's a fair amount of bare ground, that it's actually vegetation cover and productivity that's driving the variation in soil microbial processes and soil nutrient availability, more than vegetation chemistry or functional group composition, and that diversity itself is driving some of the variation in vegetation cover and productivity. At Wood River, we thought, well, there you have very high productivity, vegetation cover is 100%. Here, the variation in the vegetation chemistry and the functional group composition is likely to be a more important driver in the variation that you see, an explanatory factor in the variation we see in soil microbial processes and soil nutrient availability. So those are our working hypotheses. And this is just showing in the BioDiv experiment in Wood River, the different functional groups, which are themselves actually phylogenetic lineages, and the C3 and C4 grasses are nested within the monocots, the forbs and the legumes are nested within dicots in these experimental systems. And that's true for both BioDiv and Wood River. This is their vegetation chemistry or function, leaf mass per area, nitrogen, lignin, cell solubles, hemicellulose, and cellulose. And you can see big differences between the monocots and the dicots here, and between the functional groups. And basically, what we found in this system is that productivity, the amount of inputs, vegetation cover and biomass, which we could remotely sense, was driving the soil processes, accumulative respiration in the soil, hydrolytic enzyme activity, microbial biomass carbon, net nitrogen mineralization rates, soil carbon. But that in the Wood River system, it was the chemical variation and the functional phylogenetic composition that was driving those same things. And so remotely sensed nitrogen concentration was negatively related to oxidative enzyme activity, but positively associated with hydrolytic enzyme activity. positively predicted microbial biomass carbon and net nitrogen mineralization rates and soil carbon. And C4 composition, which is tightly correlated with cellulose, and also cellulose itself, was negatively associated with those soil processes and hydrolytic enzyme activity, microbial biomass carbon, and nitrogen minerals, net nitrogen mineralization rates, and soil carbon. So as we had predicted, the total inputs was more important in Biodiv, and the chemical composition was more important in Wood River. And this is something that we could remotely sense. And so once we had an understanding of how each system was different, then remotely sensed information could be quite valuable in predicting what was going on below ground. But it's quite important to actually understand how each of those different systems functions. In Biodiv, the productivity of the remotely sensed vegetation cover also strongly predict the community composition of the bacteria and the fungi below ground, and weakly predicted their diversity. And we just had this information for Biodiv, we didn't have it for Wood River. The next study I'll talk about is led by Laura Williams. She's a former PhD student that co-advised with Peter Reich, and also a postdoc that we co-advised on this particular project. And she has been working in one of the tree diversity experiments established at Cloquet, the IDENT experiment that's part of a series of experiments in North America and Europe with these different tree species. And they were planted in a design with varying levels of diversity, not unlike the Biodiv experiment at Cedar Creek, but with tree species now. And this is what the experiment looks like over time. And the analyses that are from Laura's paper that has just come out are from the 5th and 6th growing season. So this is a visual representation of the airborne imagery from that experiment, and the imagery, the pixels were about 1 meter or 0.8 meter pixel resolution. And the spectra looked something like this. Here's Laura. And there were a series of monocultures that could be used, the spectra from those monocultures could be used to simulate mixtures based on either the actual abundance, the way they were planted, or the way the canopies actually presented themselves at the top of the canopy, and how the airborne imagery was looking at them. And those simulated mixtures could be compared to the actual mixtures themselves, to the spectra from those actual mixtures, to capture the ecosystem consequences of the productivity effect of growing multiple species together. So that's often called the net biodiversity effect. And in general, many experiments are showing that when you grow multiple species together, you get more productivity than expected from what you get from growing individual species with each other in monocultures. And so using those spectra from monocultures, simulating what mixtures would look like if they grew the same way in mixture as they do in monoculture, and then comparing those to the observed spectra in mixtures, she was able to show, using remotely sensed information, this evidence for the spectral biodiversity effect. And the actual measured biodiversity effect on the ground, she was able to predict from remotely sensed spectra. And that depended on having a very nice model for biomass. She was able to very accurately predict biomass using a PLSR model in this system. And she was then further able to determine, using the simulation approach, whether the biodiversity effect was a consequence of dominance or plasticity. Some interesting things she found relate to nitrogen. And so first, she was able to accurately predict canopy nitrogen in this system. So this is the spectrally predicted canopy nitrogen from her models, and it accurately predict field measure canopy nitrogen. And then she was able to show that the mixtures had more nitrogen in them than you would expect from the monocultures. And that provides, her analysis of that provided a nice explanation for why you get this over yielding or this higher productivity than expected. It seems that the mixtures are taking more nitrogen out of the ground and are able to put it into their leaves, and that may contribute to their higher productivity in mixture than when species are grown individually in monoculture. And so here is looking at the spectrally detected net biodiversity effect on canopy nitrogen, and that's tightly correlated to, or it's correlated to the field measured net biodiversity effect. So those simulations required having some knowledge of which species were planted and their abundances so that she could do the simulations. And so we wanted to know, could you actually remotely sense the diversity and the productivity if you didn't know how individuals were planted in the experiment? And so using a spectral library that she developed from the experiment, she then detected species and their diversity levels without taking into account any information about how they were planted. And so this is just showing how she was able to detect individual species, monocultures based on the spectral library, and able to detect combinations of species when they were growing together. And there were some errors. And birch was good at overtopping its neighbors, and so you couldn't see them, and so that those kind of errors occurred. Or sometimes a pixel might be mixed, and so you couldn't differentiate species. Or sometimes species were so phylogenetically similar that you couldn't differentiate them. But for the most part, so there were errors, but it worked actually really well to get a measure of which were the monocultures and what was the diversity and composition of all the plots. And using that approach without any prior knowledge of the abundance and identities of species in those different plots, she was able to use a different method to spectrally predict over yielding and this net biodiversity effect. This is really quite important because if this becomes possible at landscape scales, we can solve a really critical mystery that's very important in ecology of what is the consequence of biodiversity at different spatial scales and in different systems. And it hasn't been possible to do this in natural systems over large spatial scales until now with remote sensing information, hyperspectral capabilities. So this is a big advance, and will have far-reaching implications for global understanding of the consequences of biodiversity for ecosystems. Okay, so the last part of this talk is using spectral information and remotely sensed spectral information to detect plant disease. And I've worked on oaks for the last 2 decades of my career, and I'm very concerned about the oak wilt pathogen. It's a fungal pathogen that spreads through the roots of oak trees and rapidly can kill them depending on their susceptibility. And this is in collaboration with Jenny Jeswick, Rebecca Montgomery, and John Gammon, Phil Townsend, and Anna Yang, Kathleen Lapidat, Anna Schweiger. Beth Fallon has done part of this, former postdoc, and Gerard Sapes is the postdoc who's leading this project. and it's funded by the Minnesota Invasive Terrestrial Plants and Pest Center, as well as there's funding coming from other sources as well. So oak wilt, Bretziella phagocerum, is actually the most lethal threat to oaks in the U.S. And you can see all the places, this is from the U.S. Forest Service, all the places, all the counties it's been detected in the United States. It kills the trees, it kills red oaks within a year or two, and forms these fungal mats, these sweet smelling fungal mats. And this is an image over Cedar Creek Ecosystem Science Reserve, where you can see these pockets of oak wilt where the infection spreads from one tree to the next, rapidly killing those trees. It can be spread longer distance by beetles, but that's slower than the actual spread from tree to tree below ground. If it can be detected early, it can be treated through trenching, trenches dug deep, severing the connections between neighboring species and surrounding individuals, which is something that Jenny Jeswick and her group have been working on for a long time. And the idea is that detecting it early gives you a leg up in being able to treat it, and you can use cheaper methods of treating it if it isn't so widespread in a particular forest. I want to make the point, going back to phylogenetic information, that even for detecting something like oak wilt, being able to differentiate phylogenetic lineages is really helpful for detecting the disease in this case. And so without going too much into the physiology of the disease, it is important to note that the immune systems of the two major lineages of oaks, the white oaks and the red oaks, bur oak is an example of a white oak, pin oak, northern pin oak is an example of a red oak. Their immune systems respond to the movement of the fungal pathogen in the xylem vessels quite differently. And the white oaks seem to be quite targeted in their response, quite effective and efficient in stopping the movement of the fungi through the conduits and in blocking off the infected conduits so that they don't spread to neighboring conduits. The red oaks seem to be inefficient at doing this, and they do form tyloses, but they do it too much too late. And they end up causing wilt by blocking their ability to move water to transport through the xylem, because they blocked it off in inefficient and sort of overzealous response to the fungus once it's actually too late. And so their immune systems are not good at blocking the spread of the fungus within the tree as that fungus is moving from the roots up through the stem and into branches and so forth. So the red oaks are much more susceptible than the white oaks. And they're, in eastern deciduous forests, about half the species are white oaks and half the species are red oaks. Here's an image of Quercus ellipsoidalis and its crown that has died with its neighbors still alive. Here's Quercus macrocarpa, the bur oak, and here you just see a wilted branch, but neighboring branches are still alive, and so it's able to block the spread of the pathogen between branches, and so it can actually live for quite a long time. So it has a much more efficient immune system. So being able to so that information is actually important when we're trying to detect oak wilt. And we've been working at Cedar Creek to develop models using airborne imagery to detect oak wilt. And we do this by developing spectral libraries where we have individuals that are georeferenced, and we know the identities, and we know which species they are. We know if they're oaks, we know if they're healthy or not. And we're applying this approach not only to Cedar Creek, but also to other plots that our team has been setting up around the state. And so the first thing we do is we say, is is this an oak or not an oak? And can we use spectral information to differentiate oaks from non-oaks? And as I alluded to earlier, yes, we can do that with pretty darn high accuracy, about 81% accuracy. And then within the oaks, can we differentiate red oaks from white oaks? And yes, with high accuracy, we can tell the difference between a red oak and a white oak. And then that isolates the problem to be differentiating healthy from diseased oaks. And we've done a series of experiments at the leaf level in greenhouses and outside. What I'm going to show you now, though, is just from canopies in the field. And part of what we were trying to figure out is how much does the shortwave infrared really matter? Because the sensors that use the shortwave infrared information are more expensive. Is there enough information in the visible and near infrared? And how much better are the models when we just try to detect oak wilt using a model just looking for oak wilt versus when we do this stepwise phylogenetic approach where we first find the oaks and then we differentiate the red and the white oaks and then the healthy and the live. And these are results from Gerard Sapé's work showing that, well, when you just do the direct approach, it does actually help to have the shortwave infrared, and you get higher detection of other species. But we don't do that well in detecting diseased from non-diseased. A lot of the times you actually get it wrong when you just have the veneer. You get it correct more often when you have the shortwave infrared, but still this accuracy is not as high as we'd want it. When we do this stepwise approach where we first look for oaks, then do we have a red oak, then is it diseased? Now we have pretty high accuracies in detecting the disease and somewhat higher when we use the shortwave infrared. So this is what we're finding at this point. The same information is also really useful in detecting the physiological processes as they progress through the disease. And this is using naturally occurring seedlings in the field that were infected with oak wilt. And through the progression of the disease shown in red compared to green. This is decline in water content in the canopies, actually measured leaves and then predicted from the canopies. And this is midday water potential measured versus what was predicted from the canopies. And then this is quantum yield of photosynthesis measured at the leaf level in the diseased and healthy plants. And this is measured at with spectra from the canopy using indices of reflectance that are related to chlorophyll fluorescence. And so using indices, which often are just taking into account 2 or sometimes 3 wavelengths that are linked to features related to photosynthetic function or water content or concentration of a particular chemical in the leaf. These different indices can differentiate healthy from oak wilted plants. And I'm showing here, I'll start at the leaf level here, using wavelengths represented here in this spectrum. You can differentiate oak wilt at the leaf level for this index, and using these 2 wavelengths for this one, and this index differentiates the disease using these wavelengths, and then the blue, these 2 wavelengths here. So those set of wavelengths can differentiate healthy and diseased trees at the leaf level. They also do so at the canopy level using remotely sensed imagery or spectra. In this case, it was a proximal remote sensing method. In this case, with airborne imagery, those also were able to differentiate healthy and oak wilted leaves. And then in our simulations, we haven't actually done this with real satellite data, but in our simulations of satellite data, we also believe we should be able to differentiate healthy and oak wilted plants, but that's something we're testing. And each of these indices is related to actual physiological function measured in other ways, like photosynthetic electron transport rates or light use efficiency or water potential or water content. So these indices that we're measuring that differentiate healthy and diseased plants are related to physiological functions that are changing as the disease progresses. And so that connection to the physiological function of the plants as the disease progresses allows us to scale between leaves and trees, landscapes and regions, which is what we're aiming for in this project, so that we can get efficient detection of oak wilt to make the management process less expensive and more effective. And so I think I'll wrap up here, and I'm glad to take questions about this later this week. But I wanted to mention that I'm now directing a National Science Foundation Biology Integration Institute that's using spectral biology to look at the causes and consequences of biodiversity in a rapidly changing world. And we're really asking how life's variation at each biological scale, how does it influence processes at all other scales up to the biosphere? And how do global changes influence biological variation at all of these scales? So if you want to be in contact about that, I'm glad to share more information. And finally, I'll just leave you with information about this book that came out this year, Remote Sensing of Plant Biodiversity, that I edited with John Gammon and Phil Townsend. And there's authors from across this community of people using spectral information to understand plant function and biodiversity. And it's open access, so all the chapters are freely downloadable. I'll stop there. Thanks very much.